Hello guys, and welcome to our video on cellular respiration. So first of all, let's start with an overview. So cellular respiration is a process, a method by which the cells break down glucose to generate ATP. Now, as you know from the past videos, ATP is the energy currency of the cell. And so cellular respiration is a catabolic pathway. In other words, it breaks down this larger molecule glucose into smaller molecules to generate ATP, or energy that can be used to generate ATP. Now there's an overall equation, and it goes C6H12O6, which is glucose, plus 6 molecules of oxygen results in 6 molecules of CO2 plus 6 molecules of water plus energy. So this energy is then used to make ATP. And the remainder of the energy that is not used to make ATP is dissipated as heat into the surroundings. However, it's often easy to think that this process occurs in one step, kind of like most other chemical reactions. But in reality, this doesn't occur in one step at all. Because think about it, if it happened in one step, then all this energy, all this explosive energy would release all at once, and that's really bad. Think about it like a car, for example. If your gasoline suddenly ignited all at once instead of that energy being released in several steps, then the car wouldn't go very far. It's kind of like that with cellular respiration. The energy has to be broken down step by step in order for work to actually get done. Now, the way that the energy is actually made is through the movement of electrons. So electrons are pulled away from glucose, and then they're passed down to NAD+, which is a electron carrier, and it also goes along with the proton. So this results in NADH, and this NADH is then sort of like a storage molecule in cellular respiration, and this drops off the electrons into the electron transport chain, which helps create lots of ATP. Now, 2 important terms you may want to know about are reduction, which is just simply the gain of an electron by a molecule, and oxidation, which is a loss of an electron. Alright, so let's talk about the first step of cellular respiration, which is glycolysis. So in glycolysis, what happens is the glucose is essentially broken down into 2 molecules of pyruvate. Now, pyruvate is a 3-carbon compound. So what happens is 2 ATP are broken down at the beginning, and 1 phosphate from each of the 2 ATP is added to the various intermediates in the reaction. And as you know from our past video, phosphate groups tend to increase reactivity. And so when you increase reactivity, that allows for more energy to be eventually extracted. So ultimately what happens is glycolysis is going to produce 2 ATP on net. That means that the 2 ATP that are invested are like -2, and then you add 4 ATP through the process. So on net, you're going to have 2 ATP, 2 NADH, 2 water molecules, and 2 protons are going to be made per molecule of glucose that undergoes this process. Now, this occurs regardless of the presence of oxygen. So I think this is very important because the various other steps in the process depend on whether or not there's oxygen involved. And also keep in mind that glycolysis occurs in the cytosol of the cell. Alright, so let's talk about what happens if oxygen is not present. So if oxygen is not present, glycolysis will occur, as I just explained. And what will happen after that is fermentation. So like I said, fermentation is used when there's lack of oxygen. And the reason that fermentation is used is it allows regeneration of NAD+. Now, if there is oxygen, then what you'll realize later on is that this NAD+ can be regenerated because the NADH drops off its electrons at the electron transport chain, and then the NAD+ is recreated. However, when there's no oxygen, this can't really happen. And so what happens is two molecules, mainly ethanol or lactate, are products that our NAD is going to oxidize. In other words, give electrons to regenerate NAD+. Now, notice that the pyruvate itself, or some modification of it, is going to be the final electron acceptor in fermentation instead of oxygen. Now, lactic acid fermentation, fermentation that uses lactate, is often done in human muscle cells when oxygen is used. So athletes that undergo heavy exercise are going to start using lactic acid fermentation, and that can be really painful. But ethanol fermentation is often used to like make certain alcoholic beverages, and is also used by yeast. Now let's move on to the citric acid cycle. So after glycolysis, if oxygen is present, then the citric acid cycle will occur. This is also known as the tricarboxylic cycle or the Krebs cycle. So in the citric acid cycle, pyruvate is oxidized again to form acetyl-CoA, which is a coenzyme, and this releases carbon dioxide in the process. Now what happens is most of the energy created through this cycle is stored in the form of NADH or FADH2, and these are going to be used in the electron transport chain. So 1 ATP is made per turn of the cycle. Now this allows the regeneration of NAD+ and FAD+, and so this kind of regeneration is what's going to be called, it's what's going to give the name of a cycle to this cycle. So at the end of the day, 2 ATP, 6 CO2, 8 NADH, and 2 FAD2 are made in this process per glucose molecule. Alright, so let's move on to the final step of oxidative phosphorylation, also known as the electron transport chain. So what happens here is that the NADH and the FADH are going to release their electrons into the ETC. This is going to regenerate NAD+ and FAD+. So in the electron transport chain, what happens is there are various electron carriers, and they're going to start being extremely low electronegativity, and the electronegativity is going to keep increasing until we have the penultimate electron acceptor, which is oxygen. So what's going to happen is, again, they're going to increase until oxygen receives electrons, and so what's going to happen is you're going to form water when the oxygen receives its electrons, and 2 protons, which are hydrogen atoms, combine to form water. Now, this is just a quick sequence. So what happens is the first electron acceptor is flavomononucleotide, and then you have an iron sulfur protein, and then you have coenzyme Q, also known as ubiquinone, and then you have various cytochromes afterwards. So, quick note about coenzyme Q is you might see it sold as a nutrition supplement, but it's also known as ubiquinone, and it's a small hydrophobic molecule. And what's really important about it is that it is the only non-protein molecule of the electron transport chain. Now, another thing important about cytochromes is that they have a heme group with an iron atom in the center. Now, if you know anything about red blood cells and hemoglobin, there's also an iron atom in the center of that. So think of cytochromes sort of being like hemoglobin, except slightly different in its chemical composition. Now, you may be asking, how does this generate energy? Well, what happens is electron movement is going to push H+ out of the mitochondrial matrix through by diffusion across its gradient, and then so the H+ is going to have no other way to re-enter the matrix other than through this protein called ATP synthase. And ATP synthase uses this protein uses these hydrogen ions to create ATP, to unite ADP and an inorganic phosphate to form ATP. Now here's a quick diagram. So as you can see here, what's going to be happening is that from the Krebs cycle or the citric acid cycle, you're going to see the NADH and the FADH are going to come here, and then they're going to lose their hydrogen atom and their electrons. And so the H+ is going to be pumped out, and the electrons are going to be passed along this transport chain. But notice something about the FADH2. It's located initially at the complex number 2 instead of complex number 1. This is why the FADH2 produces 1/3 less energy than NADH, because it has less potential energy due to the fact that it's introduced at a later location. So this H+ is going to be pushed out, and then what's going to happen is the H+ needs to re-enter somehow, because when all this H+ is pushed out, the gradient shifts to the point where the inner membrane space has more H+ than the matrix, and as we know from diffusion, all molecules go from higher concentration to lower concentration. So the H plus is going to move from higher to lower through its only viable route, which is the ATP synthase, also marked as V on here. And then so that's going to combine ADP and inorganic phosphate to form ATP. And so keep in mind that oxidative phosphorylation produces the vast majority of all ATP created in cellular respiration. So I hope you've enjoyed our video on cellular respiration. Make sure to subscribe and like.